Statistical methods. Measures of central tendency and dispersal 1. Measures of central tendency. Measures of central tendency describe the center point or typical value of a dataset. The three main measures are a. Mean arithmetic average. The sum of all observations divided by the total number of observations. Formula. Mean equals sigma x n, where sigma x equals sum of all values, n equals number of values. Properties. Sensitive to extreme values, outliers. Can be used for both discrete and continuous data. B. Median. The middle value in an ordered dataset. If n is even, it is the average of the two middle numbers. Procedure. Arrange data in ascending, descending order. If n is odd, median equals middle value. If n is even, median equals average of two middle values. Properties. Not affected by outliers. Best for skewed distributions. C. Mode. The value that occurs most frequently in the data. Properties. There may be more than one mode, bimodal, multimodal. Suitable for categorical as well as numerical data. Relationship among mean, median, and mode. In a symmetric distribution, mean equals median equals mode. In a positively skewed distribution, mean and GT, median and GT, mode. In a negatively skewed distribution, mode and GT, median and GT, mean. 2. Measures of dispersal, variability. Dispersal refers to the spread of data around a central value. Common measures include a. Range. The difference between the maximum and minimum values. Formula. Range equals max, min. Limitation. Affected heavily by outliers. B. Interquartile range, IQR. The difference between the 75th percentile, Q3, and the 25th percentile, Q1. IQR equals Q3, Q1. Use. Give spread of the middle 50% of data, robust to outliers. C. Variance, sigma squared or S squared. The average of the squared deviations from the mean. Population variance. Sigma squared equals sigma, chi mu, squared, n. Sample variance. S squared equals sigma, chi, x squared, n1. Properties. Units of the square of the original data. Sensitive to outliers. D. Standard deviation, sigma or S. The square root of variance. Most commonly used measure of spread. Formula. Sigma equals square root, sigma, chi mu, squared, n. Interpretation. A small standard deviation indicates data is close to the mean, a large one indicates more spread. E. Coefficient of variation, CV. The ratio of standard deviation to the mean, usually expressed as a percentage. Formula. CV equals standard deviation, mean times 100. Use. Comparing variability of datasets with different units or scales. 3. Application-based insights. Mean and standard deviation are used when data are normally distributed. Median and IQR are better for skewed data or data with outliers. CV is helpful in comparing variability across different conditions or units. 4. Practice questions Q1, GATE 2020. A dataset has mean equals 20 and standard deviation equals 4. What is the coefficient of variation? Answer. CV equals 4/20 times 100 equals 20%. Q2. Which measure of central tendency is least affected by extreme values? Answer. Median. Let me know if you'd like practice problems with solutions or to proceed to the next topic, e.g., probability, hypothesis testing, etc. You said, and dispersal, ChatGPT said. Great. Let's now dive deeper into the measures of dispersal or dispersion, the second part of your topic. Measures of dispersal, dispersion. Dispersal refers to how much the data values deviate or spread out from the central tendency, mean, median, or mode. While measures of central tendency tell us the center of the data, measures of dispersal explain the variability or consistency in the central tendency tell us the center of the data, measures of dispersal explain the variability or consistency in the dataset. 1. Range definition. The simplest measure of dispersion, calculated as the difference between the maximum and minimum values in the dataset. Formula. Range equals maximum value, minimum value. Features. Easy to compute. Highly sensitive to outliers. Doesn't give any idea about the distribution of the remaining data. 2. Interquartile range, IQR definition. The difference between the third quartile, Q3, and the first quartile, Q1. It represents the middle 50% of the dataset. Formula. IQR equals Q3, Q1. Features. Robust to outliers. Better measure of spread for skewed data. Note. Q1 equals 25th percentile, Q3 equals 75th percentile. It excludes the extreme 25% on either side of the distribution. 3. Mean absolute deviation, MAD definition. The average of the absolute differences between each data value and the mean of the dataset. Formula. MAD equals sigma, chi, x, n. Features. Represents average variability. More robust than variance for some distributions. 4. Variance definition. The average of the squared differences from the mean. It shows how far each number in the dataset is from the mean. Formulas. Population variance, sigma squared equals sigma, chi mu, squared, n. Sample variance, S squared equals sigma, chi, x squared, n1. Features. Widely used in statistical modeling. Squared units make interpretation harder. 5. Standard deviation definition. The square root of the variance. It is the most commonly used measure of spread. Formula. Standard deviation, sigma equals square root variance. Features. Expressed in the same unit as the original data. Higher SD means greater variability. 6. Coefficient of variation, CV definition. A relative measure of dispersion, useful for comparing variability between datasets with different units or means. Formula. CV equals standard deviation, mean times 100. Features. Unitless. Helps in comparing the consistency of two or more datasets. 7. Relative measures of dispersion for comparison, quartile deviation, semi-interquartile range, Q3, Q1, 2. Coefficient of quartile deviation, Q3, Q1, Q3 plus Q1. Coefficient of range, max, max plus min. Applications of measures of dispersal. In biology, to compare the variability of traits like height or gene expression. In ecology, to assess population stability or species abundance. In quality control, to determine the consistency of product characteristics. Previous year question, see Applications of measures of dispersal. In biology, to compare the variability of traits like height or gene expression. In ecology, to assess population stability or species abundance. In quality control, to determine the consistency of product characteristics. Previous year question, CSIR-NET Q, CSIR-NET June 2015. Which of the following measures is least affected by extreme values? A. Range. B. Mean. C. Standard deviation. D. Interquartile range. Answer. D. Interquartile range. MCQs for practice. Q1. Which of the following is not a measure of dispersion? A. Mean. B. Range. C. Variance. D. Standard deviation. Answer. A. Mean. Q2. The square root of variances. A. Mode. B. Mean. C. Standard deviation. D. Coefficient of variation. Answer. C. Standard deviation. Let me know if you want combined summary notes for central tendency and dispersion, or if you're ready to proceed to the next statistical topic like probability, hypothesis testing, or ANOVA. You said. Probability distributions. ChatGPT said. Here's an in-depth, exam-focused lecture on probability distributions, tailored for CSIR-NET, GATE, and other competitive exams. Probability distributions 1. What is a probability distribution? A probability distribution describes how the values of a random variable are distributed. It assigns probabilities to all possible values or intervals of values the random variable can take. There are two types of probability distributions. Discrete probability distributions. Continuous probability distributions. 2. Discrete probability distributions. These are used when the random variable takes countable values, e.g., number of heads in coin tosses. A Bernoulli distribution. Models a random experiment with only two outcomes, success 1 and failure 0. Probability of success equals p, failure equals 1 minus p. Probability mass function PMF p x equals p caret x, 1 minus p caret 1 minus x, where x equals 0 or 1. Mean mu equals p variance, sigma squared equals p, 1 minus p. B. Binomial distribution. Describes the number of successes in n-independent Bernoulli trials. Each trial has success probability p. PMF. P x equals k equals c n, k asterisk p caret k asterisk, 1 minus p caret n minus k, where k equals 0, 1, 2, n. Mean, mu equals np variance, sigma squared equals np, 1 minus p. Example, number of heads in 10 coin tosses. C. Poisson distribution. Used for modeling rare events occurring in a fixed interval of time or space. Derived as a limiting case of the binomial distribution when n right pointing arrow infinity and p right pointing arrow 0 such that np equals lambda. PMF. P x equals k equals lambda caret k asterisk e caret minus lambda k, where k equals 0, 1, 2. Mean, mu equals lambda variance, sigma squared equals lambda. Example, number of mutations in a DNA strand per 1000 bases. 3. Continuous probability distributions. Used when the random variable can take any value in a range uncountable values. A uniform distribution. All values within a given range a, b are equally likely. Probability density function PDF f x equals 1 b minus a, for as less than or equal to x is less than or equal to b. Mean, mu equals a plus b, 2 variance, sigma squared equals b minus a, squared, 12. B. Normal distribution, Gaussian distribution, bell-shaped, symmetric about the mean. Most biological and natural variables follow this distribution. PDF. Fx equals 1 square root 2p squared e caret minus x minus mu squared 2 sigma squared. Properties. Mean equals median equals mode. 68% of data lies within plus or minus 1 SD, 95% within plus or minus 2 SD, 99.7% within plus or minus 3 SD. Mean, mu variance, sigma squared. C. Exponential distribution. Models the time between occurrences of events in a Poisson process. PDF. Fx equals lambda e caret minus lambda x for x is greater than or equal to 0. Mean, mu equals 1 lambda variance, sigma squared equals 1 lambda squared. 4. Key concepts in probability distributions. Probability mass function PMF used for discrete distributions. Probability density function PDF used for continuous distributions. Cumulative distribution function CDF probability that a random variable is less than or equal to a certain value. Expected value, long-run average outcome. Variance and standard deviation, measures of spread. Previous year CSIR-NET question Q, CSIR-NET Deck 2016. Which of the following is a property of a normal distribution? At the mean, median, and mode are equal b, it is skewed to the left c, the variance is always 0 d, it has two modes. Answer. At the mean, median, and mode are equal. MCQs for practice. Q1, which of the following distributions is used to model the number of times an event occurs in a fixed interval? A. Binomial. B. Normal. C. Poisson. D. Exponential. Answer. C. Poisson. Q2, in a normal distribution, approximately how much data lies within one standard deviation of the mean? A. 50%. B. 68%. C. 75%. D. 90%. 4. Key concepts in probability distributions. Probability mass function PMF used for discrete distributions. Probability density function PDF used for continuous distributions. Cumulative distribution function CDF probability that a random variable is less than or equal to a certain value. Expected value, long-run average outcome. Variance and standard deviation, measures of spread. Previous year CSIR-NET question Q, CSIR-NET Deck 2016. Which of the following is a property of a normal distribution? At the mean, median, and mode are equal b, it is skewed to the left c, the variance is always 0 d, it has two modes. Answer. At the mean, median, and mode are equal. MCQs for practice. Q1, which of the following distributions is used to model the number of times an event occurs in a fixed interval? A. Binomial. B. Normal. C. Poisson. D. Exponential. Answer. C. Poisson. Q2, in a normal distribution, approximately how much data lies within one standard deviation of the mean? A. 50%. B 68% C, 75% D, 90%. Answer, B 68%. Let me know if you'd like to proceed to hypothesis testing or get combined summary notes of all statistical methods so far. You said, binomial, ChatGPT said. Let's now explore the binomial distribution in detail, one of the most important discrete probability distributions, frequently appearing in CSIR-NET, GATE, and other competitive exams. Binomial distribution definition. The binomial distribution models the number of successes in a fixed number of independent trials, where each trial results in either success or failure, and the probability of success B remains the same in each trial. It is a discrete probability distribution. Key conditions for binomial distribution. The experiment consists of n independent trials. Each trial has only two possible outcomes, success with probability P and failure with probability Q equals 1 minus P. The probability of success P is constant in all trials. The random variable x counts the number of successes in n trials. Probability mass function PMF x equals K equals C n, K times P caret K times 1 minus P caret n minus K, where x is the number of successes. N equals number of trials. K equals number of successes, 0 is less than or equal to K is less than or equal to n. P equals probability of success. 1 minus P equals probability of failure. C n, K equals n. K, n minus K. Mean, variance, and standard deviation, mean, expected value, mu equals n times P. Variance, sigma squared equals n times P times 1 minus P. Standard deviation, sigma equals square root n times P times 1 minus P. Shape of binomial distribution, symmetrical when P equals 0.5. Positively skewed when P and LT, 0.5. Negatively skewed when P and GT, 0.5. As n increases, the binomial distribution tends to become normally distributed due to the central limit theorem. Examples. Tossing a coin 5 times. Let x be the number of heads. N equals 5, P equals 0.5, then x tilde bin 5, 0.5. A test has 10 multiple choice questions with 4 options each. The chance of guessing correctly is 0.25. If a student guesses all answers, the number of correct answers is binomial with n equals 10, P equals 0.25. Application in biology and life sciences, probability of a gene being inherited in Mendelian genetics. Number of mutated individuals in a population. Estimating probability of success failure in biological experiments, e.g., PCR, gel runs. Previous year CSIR-NET GATE question Q, CSIR-NET June 2017. If the probability of success in a binomial distribution is P equals 0.6 and the number of trials is 10, what is the probability of exactly 6 successes? Solution. P x equals 6 equals C 10, 6 times 0.6 caret 6 times 0.4 caret 4 equals 210 times 0.046656 times 0.0256 equals approximately 0.251. Answer, approximately 0.251. MCQs for practice. Q1, in a binomial distribution with n equals 5 and P equals 0.2, what is the mean? Up 1. B 0.5 C 2D 4. Answer, up 1 mean equals n times P equals 5 times 0.2. Q2, which of the following is not a condition for binomial distribution? A fixed number of trials. B independent trials C varying probability of success D only 2 outcomes for trial answer, C varying probability of success. Let me know if you'd like to move on to Poisson distribution or normal distribution next, or if you'd like problem sets with detailed solutions for binomial distribution. You said. More detailed lecture paragraph type ChatGPT said. Absolutely. Here's a detailed, paragraph-style lecture on the binomial distribution, tailored for CSIR-NET Life Sciences, GATE Ecology and Evolution, and other advanced level exams. This version focuses on both conceptual clarity and exam level depth. Binomial distribution, detailed lecture. The binomial distribution is a fundamental discrete probability distribution that describes the behavior of a binary two-outcome experiment repeated a fixed number of times. In biological research and life sciences, it is widely used to model probabilities where outcomes are dichotomous, such as survival versus death, presence versus absence of a gene, or success versus failure in an experiment. The distribution arises when a process or experiment meets specific assumptions. I. The experiment is repeated n times. E. Each trial is independent of the others. E. There are only 2 possible outcomes for trial, commonly referred to as success and failure, and IV. The probability of success P remains constant for each trial. The probability of failure is then given by Q equals 1 minus P. Let x denote the random variable representing the number of successes in n trials. Then, x follows a binomial distribution and is denoted as x tilde b n p. The probability of observing exactly k successes in n trials is given by the binomial probability mass function PMF. P x equals K equals C n, K times P caret K times Q caret n minus K. Where C n, K equals n, K, n minus K, is the binomial coefficient, representing the number of ways to choose k successes out of n trials. This function calculates the probability of a specific number of successes k occurring in a fixed number of Bernoulli trials, making it extremely useful in biological sampling, genetic probabilities, and diagnostic testing. The expected value mean of a binomial distribution is mu equals np, reflecting the average number of successes expected in n trials. The variance, which measures the spread of the distribution, is given by sigma squared equals np, 1 minus p. The standard deviation is simply the square root of the variance, sigma equals square root np, 1 minus p. These parameters help in determining how consistent or spread out the outcomes are around the mean. If the probability of success is p equals 0.5 and the number of trials is large, the binomial distribution is symmetric. However, when p is far from 0.5, the distribution becomes skewed, right skewed when p and LT, 0.5 and left skewed when p and GT, 0.5. In biological and ecological research, binomial models are frequently applied. For example, in population genetics, it models the inheritance of alleles where each gamete has a probability p of carrying a particular gene. In field ecology, binomial distribution can model the presence or absence of a species in quadrats. In molecular biology, it is used to model the success rate of PCR amplification or mutation detection in a sample of DNA sequences. In clinical trials, the number of patients responding positively to a drug out of a fixed number tested can also be modeled using binomial distribution. An important feature of the binomial distribution is its relationship with other distributions. When the number of trials becomes very large, n right pointing arrow infinity, and the probability of success is small, p right pointing arrow O, such that the mean np remains finite, the binomial distribution converges to a Poisson distribution. This is especially useful when modeling rare events. Additionally, by the central limit theorem, the binomial distribution approximates a normal distribution as n becomes large and p is not too close to 0 or 1. This makes it possible to use normal approximation methods for calculating binomial probabilities when exact computation becomes cumbersome due to large values of n. To illustrate, consider a genetic cross where the probability of an offspring being heterozygous is 0.5. In a sample of 10 offspring, the number of heterozygous individuals x follows a binomial distribution with n equals 10 and p equals 0.5. The probability of observing exactly 6 heterozygous individuals is calculated using the PMF as p x equals 6 equals c10, 6 times 0.5 caret 6 times 0.5 caret 4 equals 210 times 0.015625 times 0.0625 approximately equals 0.205. This type of calculation is often tested in CSIR-NET and GATE exams, especially in questions involving statistical modeling of biological processes. Understanding the shape, properties, and applications of the binomial distribution is thus essential for anyone dealing with biological data analysis, probabilistic modeling, or ecological predictions. Previous year question, CSIR-NET Deck 2017. A population of insects has a 25% chance of survival in a pesticide test. If 8 individuals are tested, what is the probability that exactly 2 survive? Solution. Here, n equals 8, p equals 0.25, k equals 2, p x equals 2 equals c8, 2 times 0.25 squared times 0.75 caret 6 equals 28 times 0.0625 times 0.1779785 approximately equals 0.311. Answer, 0.311. MCQs for practice. For practice. Q1, in a binomial experiment with n equals 10 and p equals 0.7, the variance is, at 2.1. B3C, 7D, 1.5. Answer, a 2.1 sigma squared equals np, 1 minus p equals 10 times 0.7 times 0.3. Q2, the binomial distribution becomes symmetric when a n is small. BP equals 0.5 CP equals 0.2D NP and LT. One answer, BP equals 0.5. Let me know if you want the next lecture on Poisson distribution, or if you need a PDF version, summary notes, or more CSIR GATE questions with solutions based on the binomial model. You said, normal ChatGPT said. Here's a detailed paragraph-style lecture on the normal distribution, written in depth and aligned with CSIR-NET Life Sciences and GATE Ecology and Evolution Preparation Standards. This lecture covers concepts, properties, biological applications, and exam-relevant insights with maximum coverage. Normal distribution, detailed lecture. The normal distribution is one of the most important and widely used probability distributions in statistics and biological sciences. It is a continuous probability distribution that describes how the values of a variable are distributed around the mean, mu. The distribution is symmetric, bell-shaped, and characterized by two parameters, the mean, mu, which determines the location or center of the distribution, and the standard deviation, sigma, which controls the spread or dispersion. In its standard form, the normal distribution is denoted as x tilde n mu, sigma squared, where sigma squared is the variance. The probability density function PDF of the normal distribution is given by the formula fx equals 1 square root 2p squared times express minus x minus mu squared 2 sigma squared. This function defines the shape of the distribution, which is symmetric about the mean. The highest point on the curve occurs at x equals mu, and the curve tails off equally on both sides, never touching the x-axis. Because of its shape, the normal distribution is often called a Gaussian distribution, named after the mathematician Carl Friedrich Gauss. A key property of the normal distribution is the empirical rule or 6895-99.7 rule. According to this rule, approximately 68% of the data falls within 1 standard deviation, mu plus or minus sigma, 95% within 2 standard deviations, mu plus or minus 2 sigma, and 99.7% within 3 standard deviations, mu plus or minus 3 sigma, from the mean. This property makes the normal distribution extremely useful for predicting the range of variation in natural phenomena. It also provides a framework for statistical inference, such as hypothesis testing and confidence interval estimation. In biological sciences, the normal distribution is used to model variables that are the outcome of multiple small, independent factors, such as height, weight, enzyme activity levels, gene expression intensities, and biomarker concentrations. Many biological traits are polygenic, meaning they are controlled by multiple genes and environmental interactions. Due to the central limit theorem, the sum or average of a large number of independent random variables tends to follow a normal distribution, regardless of the underlying distribution of each variable. This is why many complex biological traits and measurement errors tend to be normally distributed. Another important aspect is the standard normal distribution, which is a special case of the normal distribution with mean 0 and standard deviation 1. Any normal distribution can be transformed into a standard normal distribution using the z transformation, z equals x minus mu sigma. This transformation is critical for statistical tests such as z-tests, t-tests, and for computing probabilities using standard normal tables. For example, if a biological sample has a mean enzyme activity of 150 units with a standard deviation of 10, and a particular reading is 170 units, the corresponding z-score would be 170 minus 150, 10 equals 2. This tells us how many standard deviations away from the mean that reading lies, and how probable or rare such a value is under the normal model. From a graphical perspective, the normal curve is unimodal, with the mode, median, and mean coinciding at the center mu. The curve is asymptotic to the x-axis, meaning it never touches or crosses the axis. The total area under the curve is 1, which represents the total probability. One critical feature is that even though the distribution extends indefinitely in both directions, the probability of extremely large deviations becomes infinitesimally small. In ecology and evolution, the normal distribution is frequently used in trait distribution analysis, population sampling, and quantitative genetics. For instance, the distribution of beak lengths in a bird population, or weights of plant seeds in an agricultural experiment, often approximates a normal curve. In experimental biology, assumptions of normality are foundational for applying parametric tests such as ANOVA and regression analysis. Many experimental designs also rely on the normal distribution to interpret variability and estimate confidence in measurements. Previous year question, CSIR-NET Deck 2016. Q. In a normal distribution, what is the probability of obtaining a value more than 2 standard deviations away from the mean? Solution. From the empirical rule, approximately 95% of values lie within plus or minus 2 sigma. So, the remaining 5% lie outside this range. Answer. 5% or 0.05. Practice MCQs. Q1. The shape of a normal distribution is a skewed right. B. Skewed left. C. Bell-shaped and symmetric. D. Uniform. Answer. C. Bell-shaped and symmetric. Q2. In a normal distribution with mu equals 100 and sigma equals 10, what is the z-score for x equals 120? Up 1. B2 C 0 D3. Answer. B2. Let me know if you'd like the next lecture on Poisson distribution or a comparison between binomial, normal, and Poisson distributions for exam revision. Here's a detailed paragraph-style lecture on sampling distribution, tailored for CSIR-NET Life Sciences, GATE Ecology and Evolution, and related exams. It explains the concept, its mathematical foundation, significance in biological sciences, and links to statistical inference, with maximum coverage for advanced understanding. Sampling distribution, detailed lecture. The concept of sampling distribution lies at the heart of inferential statistics. A sampling distribution refers to the probability distribution of a given statistic, such as the mean, proportion, or variance calculated from a large number of random samples drawn from a population. Unlike a population distribution, which shows how individual data points are spread, a sampling distribution shows how sample statistics behave when the sampling process is repeated multiple times under identical conditions. It provides a mathematical bridge between a sample and the population, allowing researchers to estimate population parameters and test hypotheses based on sample data. Consider a biological population with a known distribution of a trait, e.g., seed weight in a plant species. If we take a random sample of size n from this population and calculate the sample mean x and then repeat this process many times, we get a distribution of these sample means. This distribution of sample means is the sampling distribution of the sample mean. One of the key results in statistics is that, regardless of the population distribution, the sampling distribution of the sample mean approaches a normal distribution as the sample size increases, a property known as the central limit theorem, CLT. This result justifies the frequent use of normal-based statistical methods even when the population is not normally distributed, especially for large sample sizes n and gt, 30. The mean of the sampling distribution of the sample mean is equal to the population mean, mu, and the standard deviation of the sampling distribution is called the standard error, SE. For the sample mean, the standard error is calculated as SE equals sigma square root n, where sigma is the population standard deviation and n is the sample size. This formula shows that larger sample sizes reduce the variability in sample means, making them more accurate estimates of the population mean. In practical terms, this means that increasing the sample size improves the precision of our statistical estimates. In biological sciences, sampling distributions are essential when conducting hypothesis testing, constructing confidence intervals, and assessing the reliability of sample estimates. For example, when estimating the average growth rate of a species under controlled lab conditions, researchers rely on sample data. The sampling distribution of the mean allows them to construct confidence intervals to capture the true growth rate and to determine whether observed differences between treatment groups are statistically significant. Similarly, in ecology, when assessing biodiversity indices across different habitats using sample plots, researchers use sampling distributions to make inferences about larger ecosystems. Different statistics have different sampling distributions. For instance, the sampling distribution of a sample proportion, e.g., the proportion of diseased plants in a crop sample, is also approximately normal for large samples and has standard error SE equals square root p 1 minus p n, where p is the sample proportion. For variance and standard deviation, the sampling distributions are not normal and often follow chi-square or F distributions, which are used in ANOVA and hypothesis testing involving variances. Importantly, the shape of the sampling distribution depends on both the population distribution and the sample size. If the population is normal, the sampling distribution of the mean will also be normal, regardless of sample size. However, if the population distribution is skewed or non-normal, a larger sample size is required for the sampling distribution to approximate normality, which is again guaranteed by the central limit theorem. This principle is critical in determining whether parametric tests, which assume normality, or non-parametric tests, which don't, should be used. Understanding the sampling distribution also explains why repeated sampling gives different results, and how much those results can vary due to chance. This insight leads to the development of p-values, confidence intervals, and statistical power, which are essential for making reliable decisions in scientific research. Previous year question, CSIR-NET December 2015 Q. What is the expected value of the sampling distribution of the sample mean? Answer. The population mean, mu. Practice MCQs. Q1. The standard error of the sample mean decreases when a sample size increases. B. Population variance increases. C. Sample size decreases. D. Mean becomes 0. Answer. A sample size increases. Q2. Which theorem explains why the sampling distribution of the mean becomes normal as sample size increases? A. Law of large numbers. B. Bayes theorem. C. Central limit theorem. D. Z-score theorem. Answer. C. Central limit theorem. Let me know if you'd like to proceed with the next lecture on t-distribution or confidence intervals, or if you'd like a combined summary of all sampling-related distributions for revision. You said. Difference between parametric and non-parametric statistics, ChatGPT said. Here's a detailed paragraph-style lecture on the difference between parametric and non-parametric statistics, tailored for CSIR-NET Life Sciences, GATE Ecology and Evolution, and other competitive exams. This explanation is structured to help in both comprehension and exam writing. Parametric versus non-parametric statistics, detailed lecture. Parametric and non-parametric statistics are two broad branches of statistical inference used for analyzing data, each with its own assumptions, applications, and strengths. The fundamental difference between the two lies in their assumptions about the population distribution from which a sample is drawn. Parametric statistics rely on the assumption that the data follow a specific distribution, most commonly the normal distribution. These methods require knowledge of certain population parameters, such as the mean, mu, and standard deviation, sigma, and are used when the underlying population is assumed to be continuous and well-defined. Examples of commonly used parametric tests include the t-test, z-test, ANOVA, analysis of variance, and linear regression. These tests are powerful and efficient when their assumptions are met, meaning they have a higher probability of detecting a true effect, statistical power. However, if the assumptions, especially normality and homogeneity of variances, are violated, the results of parametric tests may be misleading or invalid. In contrast, non-parametric statistics do not require the assumption of any specific population distribution. These tests are also known as distribution-free tests, and they are especially useful when the data are ordinal, ranked, skewed, or not normally distributed, or when sample sizes are small. Non-parametric methods are more flexible and robust in these situations, as they are based on medians, ranks, or signs, rather than means and standard deviations. Common non-parametric tests include the Mann-Whitney U-test, alternative to independent samples t-test, Wilcoxon signed rank test, alternative to paired t-test, Creswell-Wallace test, alternative to one-way ANOVA, and the chi-square test used for categorical data. While non-parametric tests are less powerful than parametric tests under ideal conditions, they are more reliable when assumptions are violated. In biological research, both approaches are important. For instance, if one is comparing the body weight of two animal populations with a large sample size and data are normally distributed, a parametric t-test is suitable. But if the data are non-normal, contain outliers, or are measured on an ordinal scale, e.g., pain score severity rank, a non-parametric Mann-Whitney U test would be preferred. In ecological field studies, where data are often skewed due to environmental variability or small sample sizes, non-parametric tests provide a safer statistical framework. Another distinction is that parametric methods allow for estimation and confidence intervals of population parameters, while non-parametric methods usually do not yield such estimates directly, though modern computational techniques like bootstrapping can be applied. Moreover, non-parametric methods are often easier to compute manually, which historically made them popular before the rise of statistical software. Ultimately, the choice between parametric and non-parametric methods depends on the type of data, sample size, research question, and whether the assumptions of parametric tests are met. It is generally advisable to test for normality, e.g., using the Shapiro-Wilk test or QQ-plot before applying parametric methods. If normality is not supported or if the data are ordinal or nominal, a non-parametric test should be used. Previous year question gave Ecology and Evolution 2020 Q. Which of the following is a non-parametric test used to compare two independent groups? A paired t-test. B. Wilcoxon-signed rank test. C. Mann-Whitney U test. D. ANOVA. Answer: C. Mann-Whitney U test. Practice MCQs. Q1. Parametric tests are most appropriate when A. Data are ordinal and non-normal. B. Sample size is very small. C. Data are categorical. D. Data are continuous and normally distributed. Answer: D. Data are continuous and normally distributed. Q2. Which of the following is not a parametric test? A. One-way ANOVA. B. T-test. C. Chi-square test. D. Linear regression. Answer: C. Chi-square test. Let me know if you'd like the next lecture on types of errors, type 1 and type 2, or statistical power and sample size estimation. You said. Confidence interval, ChatGPT said. Here's a comprehensive, paragraph-style lecture on confidence intervals, written for CSIR Net Life Sciences and GATE Ecology and Evolution level preparation. This lecture covers the theory, intuition, mathematical logic, biological applications, and past exam relevance. Confidence interval, detailed lecture. A confidence interval, C, is a range of values, derived from sample data, that is likely to contain the true value of an unknown population parameter, such as the mean, proportion, or difference between means. Rather than giving a single estimate, confidence intervals provide a margin of error and reflect the degree of uncertainty or confidence associated with the sample estimate. The most commonly used confidence level is 95%, which means that if we were to take 100 different samples and compute a confidence interval from each, approximately 95 of them would contain the true population parameter. Thus, confidence intervals are a fundamental part of inferential statistics, allowing scientists to generalize sample findings to the larger population while acknowledging variability. The general form of a confidence interval for the population mean mu, when the population standard deviation sigma is known and the sample size is large and is greater than or equal to 30, is given by c equals x plus or minus c asterisk sigma square root n. Where x is the sample mean, z is the z-score corresponding to the desired confidence level, e.g., 1.96 for 95%, sigma is the standard deviation of the population, and n is the sample size. When the population standard deviation is unknown and the sample size is small, n and LT, 30, we use the t distribution instead of the z distribution, and the formula becomes c equals x plus or minus t asterisk s square root n. Where s is the sample standard deviation, and t is the t-score based on the degrees of freedom, n1. In biological sciences, confidence intervals are essential in expressing precision and reliability of estimates. For example, in a clinical trial comparing two treatments, a 95% confidence interval for the difference in mean recovery times might be -1.2, 3.6 days. This interval suggests that while the new treatment might be faster, the range includes 0, indicating that the difference might not be statistically significant. Similarly, in ecology, confidence intervals are used when estimating biodiversity indices, population densities, or enzyme activity levels from sample data, providing a statistically defensible range rather than a fixed estimate. The width of a confidence interval depends on several factors. Sample size. Larger samples yield narrower intervals due to reduced standard error. Variability in data. Greater variability, standard deviation, results in wider intervals. Confidence level. Higher confidence, e.g., 99%, leads to wider intervals, since we want to be more certain that the interval captures the true parameter. It is important to interpret confidence intervals correctly. A 95% confidence interval does not mean there's a 95% probability that the population parameter lies within the calculated interval from a single sample. Instead, it means that over many repeated samples, 95% of the constructed intervals will contain the parameter. This subtle distinction is key in avoiding misinterpretation of statistical results. Confidence intervals also help in hypothesis testing. For instance, if a 95% C for a mean difference excludes 0, it suggests the difference is statistically significant at the 0.05 level. They are thus widely preferred over p-values alone, as they convey more information, both the significance and the magnitude of the effect. In modern biology and ecology, reporting confidence intervals has become standard practice in journals and experiments. It ensures transparency, reproducibility, and allows for better meta-analysis and cross-study comparisons. Previous year question, CSIR Net Deck 2014. Q. A 95% confidence interval of 1.5, 3.5 for the mean length of a fish species suggests that the population mean is 2.5 centimeters. B. 95% of fish are between 1.5 and 3.5 centimeters. C. There is a 95% probability that the true mean lies between 1.5 and 3.5 centimeters. D. Repeated sampling would yield intervals containing the true mean 95% of the time. Answer. D. Repeated sampling would yield intervals containing the true mean 95% of the time. Practice MCQs. Q1. Which of the following increases the width of a 95% confidence interval for the mean? A. Increasing sample size. B. Decreasing population standard deviation. C. Using a 99% confidence level instead. D. Increasing the sample mean. Answer. C. Using a 99% confidence level instead. Q2. A 95% confidence interval for the mean height of a plant species is 35 centimeters, 45 centimeters. Which statement is correct? A. All plants fall within this range. B. The mean of the sample is 40 centimeters. C. The population mean is definitely between 35 and 45 centimeters. D. 95% of future samples will lie in this range. Answer. B. The mean of the sample is 40 centimeters. Let me know if you'd like the next lecture on type 1 and type 2 errors, t-distribution, or p-values and hypothesis testing. You said, errors, ChatGPT said. Here's a detailed, paragraph-style lecture on type 1 and type 2 errors, essential for CSIR Net Life Sciences, GATE Ecology and Evolution, and other competitive exams. This explanation includes conceptual clarity, real-life biological examples, and exam-focused insight. Type 1 and type 2 errors, detailed lecture. In hypothesis testing, we assess whether to reject or fail to reject a null hypothesis H based on sample data. However, since we rely on probabilistic reasoning and sample evidence, there's always a possibility of making errors. These errors are broadly classified into type 1 and type 2 errors, and understanding them is crucial for interpreting the outcomes of statistical tests in biological and ecological research. A type 1 error occurs when we reject the null hypothesis H when it is actually true. In other words, we mistakenly conclude that there is an effect or difference when none exists in the population. This is also called a false positive. The probability of committing a type 1 error is denoted by the Greek letter alpha, which represents the significance level of the test. Common values for alpha are 0.05 or 0.01, meaning there's a 5% or 1% chance of incorrectly rejecting H. In biological research, a type 1 error might occur if we conclude that a new drug is effective when in fact it is not. Minimizing type 1 errors is important in contexts where false claims can have serious consequences, such as in clinical trials or conservation biology. A type 2 error, on the other hand, occurs when we fail to reject the null hypothesis when it is actually false, that is, we miss detecting a real effect or difference. This is called a false negative, and the probability of making a type 2 error is denoted by beta. The power of a statistical test is defined as 1 beta, and it reflects the test's ability to detect a true effect when it exists. Increasing sample size, effect size, and significance level alpha can help reduce type 2 errors and increase statistical power. For example, in ecological studies, a type 2 error might occur if we fail to detect the effect of pollution on species richness even though the effect is real, possibly due to insufficient sample size or high natural variability in the data. Balancing type 1 and type 2 errors is a fundamental trade-off in hypothesis testing. Lowering alpha reduces the chance of type 1 errors but increases the risk of type 2 errors, and vice versa. The choice of alpha thus depends on the context and consequences of the error. In medical research, we may tolerate a small risk of type 1 error but prefer to minimize type 2 error to avoid missing potentially life-saving treatments. In contrast, in toxicology or conservation decisions, avoiding false positives may be prioritized to prevent unnecessary actions. Let's consider a biological example. A study tests whether a certain pesticide causes mortality in a frog population. Type 1 error, conclude the pesticide causes mortality when it actually doesn't, false alarm. Type 2 error, fail to detect pesticide-induced mortality when it actually exists, missed risk. Each of these errors can lead to incorrect policy or conservation decisions. Understanding these errors is vital for interpreting p-values. A p-value less than alpha leads us to reject H, accepting a small probability alpha that this is a type 1 error. However, a non-significant result doesn't prove H is true. It might reflect low power or small effect size, leading to a type 2 error. Previous year question, GATE Ecology and Evolution 2021. Q. In a hypothesis test, if a null hypothesis is true but is rejected, which of the following errors has occurred? A type 1 error. B. Type 2 error. C. No error. D. Both errors. Answer. A type 1 error. Practice MCQs. Q1. Which of the following increases the risk of type 2 error? A. Increasing sample size. B. Increasing effect size. C. Decreasing alpha, significance level. D. Increasing power. Answer. C. Decreasing alpha, significance level. Q2. The probability of committing a type 1 error is represented by A beta. B. 1 alpha. C. Alpha. D. 1 beta. Answer. C. Alpha. Let me know if you'd like the next lecture on statistical power, p-values and hypothesis testing, or t-distribution and degrees of freedom. You said. Levels of significance, ChatGPT said. Here's a detailed, paragraph-style lecture on levels of significance, written in an exam-oriented manner suitable for CSIR Net Life Sciences and GATE Ecology and Evolution. This lecture provides the theoretical core, mathematical interpretation, biological relevance, and exam application. Levels of significance, detailed lecture. The level of significance, commonly denoted by the Greek letter alpha, is a critical concept in hypothesis testing. It represents the probability of rejecting the null hypothesis H when it is actually true, i.e., the risk of making a type 1 error. In simpler terms, it defines how much uncertainty or tolerance for error a researcher is willing to accept when drawing conclusions from statistical data. The level of significance is set before performing a test and guides the interpretation of the p-value obtained from the analysis. The most commonly used levels of significance in scientific research are 0.05, 5%, 0.01, 1%, and 0.10, 10%. An alpha of 0.05, for example, means that there is a 5% chance of falsely rejecting a true null hypothesis, a false positive result. If the p-value obtained from a statistical test is less than or equal to alpha, we reject the null hypothesis, considering the result statistically significant. If the p-value is greater than alpha, we fail to reject the null, suggesting that any observed difference could be due to chance. The choice of significance level depends on the context of the study and consequences of error. In medical or toxicological studies where a type 1 error could lead to harmful consequences, e.g., approving an unsafe drug, researchers may use a stricter alpha like 0.01. In ecological fieldwork or exploratory biological studies where a more lenient threshold is acceptable, 0.05 or even 0.10 may be used. It's important to note that reducing alpha decreases the chance of type 1 error but increases the chance of type 2 error, beta, potentially missing a true effect. Thus, a balance between alpha and beta must be achieved based on the scientific objective. Levels of significance are also directly related to confidence levels. For example, a significance level of 0.05 corresponds to a 95% confidence level, meaning we are 95% confident that our statistical conclusion is correct. In the case of a two-tailed test, alpha is divided into two equal parts, with each tail of the distribution having alpha 2. This is done when we test for any significant difference, regardless of direction. In biological sciences, significance levels are applied in evaluating experimental results, such as comparing mean growth rates, enzyme activities, population densities, or survival rates between control and treatment groups. For example, in a study testing whether a nutrient supplement increases plant biomass, a p-value of 0.03 would be considered statistically significant at alpha equals 0.05, and we would conclude that the supplement has a real effect on growth. It is important to interpret the significance level with caution. A statistically significant result does not always imply biological or practical importance. Conversely, a non-significant result does not always mean no effect exists. It might result from small sample sizes or high variability. Hence, effect size, confidence intervals, and replication should be considered alongside alpha for robust conclusions. Previous year question CSIR Net Deck 2018 Q. A researcher sets the level of significance at 0.01. What does this imply? A. There's a 1% chance of accepting a false hypothesis. B. There's a 1% chance of making a type 1 error. C. There's a 1% chance of making a type 2 error. D. The confidence level is 99%. Answer. B. There's a 1% chance of making a type 1 error. Practice MCQs. Q1. A test is conducted at the 5%. A. There's a 1% chance of accepting a false hypothesis. B. There's a 1% chance of making a type 1 error. C. There's a 1% chance of making a type 2 error. D. The confidence level is 99%. Answer. B. There's a 1% chance of making a type 1 error. Practice MCQs. Q1. A test is conducted at the 5% level of significance. Which of the following is true? A. The test will not make any error. B. The chance of making a type 1 error is 0.05. C. The chance of making a type 2 error is 0.05. D. The p-value must be exactly 0.05. Answer. B. The chance of making a type 1 error is 0.05. Q2. What happens if we decrease the level of significance from 0.05 to 0.01? A. We become more likely to detect a true effect. B. Type 2 error increases. C. Type 1 error increases. D. Confidence level decreases. Answer. B. Type 2 error increases. Let me know if you want the next lecture on p-value and hypothesis testing, or any other topic from your statistics syllabus. You said, regression. ChatGPT said. Here's a detailed, paragraph-style lecture on regression analysis, designed especially for CSIR Net Life Sciences, GATE Ecology and Evolution, and other postgraduate level competitive exams. The explanation includes theoretical depth, application in biological sciences, and related practice questions with answers. Regression analysis, detailed lecture. Regression analysis is a statistical method used to examine the relationship between a dependent variable and one or more independent variables. It helps predict the value of the dependent variable based on the known values of the independent variables, and is widely used in biology, ecology, and life sciences to analyze experimental and observational data. The most commonly used form is simple linear regression, which models the relationship between two continuous variables using a straight line. When more than one predictor is involved, it is called multiple regression. In simple linear regression, we study the relationship between a single independent variable x and a dependent variable y by fitting a regression line of the form y equals a plus bx plus epsilon. Where y is the dependent variable, x is the independent variable, a is the intercept value of y when x equals o, b is the slope or regression coefficient indicating the rate of change in y per unit change in x, and epsilon is the random error term. The slope b is calculated in such a way that the sum of the squared differences between the observed and predicted y values, i.e., residuals, is minimized. This method is called the least squares method. The regression coefficient b indicates the direction and strength of the relationship. A positive slope indicates a direct relationship, y increases with x, while a negative slope indicates an inverse relationship, y decreases with x. If b equals 0, there is no linear relationship. In biological studies, regression is useful for predicting values and understanding causal or correlational relationships. For example, researchers might use regression to predict body mass from bone length invertebrates, or to assess how temperature affects enzyme activity. In ecological studies, regression can help model how species richness changes with altitude or pollution levels, or how primary productivity is influenced by rainfall. Regression analysis also provides important metrics such as the coefficient of determination, r squared, which represents the proportion of variance in the dependent variable explained by the model. An r squared of 0.85 means that 85% of the variation in y is explained by x. However, a high r squared does not imply causation, it only suggests a strong association. It's important to distinguish correlation from regression. Correlation measures association and direction between two variables, without distinguishing between dependent and independent variables. Regression, on the other hand, predicts or explains how one variable affects another, with a clear distinction between predictor x and response y. Regression assumes certain conditions. 1. Linear relationship between variables. 2. Homoscedasticity, constant variance of errors. 3. Independence of observations. And 4. Normally distributed residuals. If these assumptions are violated, the regression model may produce biased or unreliable results. In such cases, nonlinear regression or non-parametric methods may be used. In multiple regression, we extend the model to include multiple predictors, y equals a plus bx plus bx plus bx plus epsilon. This is useful in complex biological systems where multiple factors jointly influence the response variable, for example, predicting plant growth based on sunlight, water, and nutrient availability. Previous year question, GATE Ecology and Evolution 2020. Q. In a simple linear regression analysis, the regression coefficient slope was found to be positive. What does this indicate? A. The dependent variable increases with the independent variable. B. The dependent variable decreases with the independent variable. C. No relationship exists between the variables. D. The variance of y is negative. Answer. A. The dependent variable increases with the independent variable. Practice MCQs. Q1. The coefficient of determination r squared in a regression model indicates a. The correlation between two variables. B. The significance level of a model. C. The proportion of variance in y explained by x. D. The slope of the regression line. Answer. C. The proportion of variance in y explained by x. Q2. If a regression model has a slope of 0, what does it mean? A. X and y are perfectly correlated. B. Y decreases with x. C. Y does not change with x. D. The model is nonlinear. Answer. C. Y does not change with x. Let me know if you want the next lecture on correlation analysis, multiple regression, or ANOVA analysis of variance. Here's a detailed, paragraph-style lecture on correlation, tailored for CSIR Net Life Sciences, GATE Ecology and Evolution, and similar postgraduate level exams. This lecture includes core theory, interpretation, biological examples, and practice questions with answers. Correlation, detailed lecture. Correlation is a statistical technique used to measure and describe the strength and direction of the relationship between two quantitative variables. It helps answer the question, do two variables move together, and if so, how strongly and in what direction? The most widely used measure of correlation is the Pearson correlation coefficient, denoted as R, which quantifies the degree of linear association between two continuous variables. The Pearson correlation coefficient R ranges from -1 to +1. A value of +1 indicates a perfect positive linear relationship, meaning that as one variable increases, the other increases proportionally. A value of -1 indicates a perfect negative linear relationship, meaning that as one variable increases, the other decreases proportionally. A value of 0 indicates no linear relationship between the variables. Values closer to plus or -1 signify stronger relationships, while values near 0 imply weak or no linear association. Correlation is symmetric, meaning the correlation of x with y is the same as that of y with x. Unlike regression, correlation does not imply causation and does not distinguish between dependent and independent variables. It simply assesses how two variables covary, without assuming one affects the other. In biology and ecology, correlation is often used in observational studies where controlled experiments are not feasible. For example, researchers may examine the correlation between soil nitrogen levels and plant species richness, or between body length and metabolic rate in lizards. These relationships help formulate hypotheses but require further testing for causality. Spearman's rank correlation coefficient, ρ or RS, is another measure used when data are ordinal, non-parametric, or not normally distributed. It assesses monotonic relationships based on the ranks of the data rather than their raw values. This is particularly useful in biological studies involving qualitative or ranked data, such as dominance hierarchies in animals or preference rankings in behavioral assays. It is important to visualize data using scatter plots before calculating correlation, to ensure the relationship is approximately linear and not driven by outliers. In the presence of nonlinear patterns, Pearson's R may be misleading even if the variables are related. Also, spurious correlations can occur due to confounding factors or coincidence, especially in large datasets. Careful interpretation is crucial. The coefficient of determination R squared, derived from the square of the correlation coefficient, indicates the proportion of variability in one variable that is explained by the other. For instance, if R equals 0.8, then R squared equals 0.64, meaning 64% of the variability in Y is explained by X, and the remaining 36% is due to other factors. Previous year question, CSIR Net June 2017. Q. Two variables X and Y have a Pearson correlation coefficient R equals -0.95. What does this imply? A strong positive correlation. B. No relationship between X and Y. C. A strong negative linear correlation. D. Y increases as X increases. Answer. C. A strong negative linear correlation. Practice MCQs. Q1. Which of the following statements about correlation is true? A. Correlation implies causation. B. Correlation is asymmetric. C. Correlation measures the linear association between variables. D. Pearson correlation is used for categorical data. Answer. C. Correlation measures the linear association between variables. Q2. If the correlation coefficient between two variables is 0, it means that the variables are independent. B. The variables have a strong linear relationship. C. The variables have no linear relationship. D. There is a perfect triple linear relationship. Answer. C. The variables have no linear relationship. Let me know if you want to continue with ANOVA analysis of variance or go deeper into Spearman's correlation, partial correlation, or other statistical tests next. You said, t-test, ChatGPT said. Here's a detailed, paragraph-style lecture on the t-test, designed for CSIR Net Life Sciences, GATE Ecology and Evolution, and other postgraduate level exams. This includes essential theory, assumptions, biological examples, and exam-focused questions. T-test, detailed lecture. The t-test is a parametric statistical test used to determine whether there is a significant difference between the means of two groups. It is especially useful when dealing with small sample sizes, typically N and LT, 30, and is widely applied in biological, ecological, and behavioral sciences to compare group responses under different experimental conditions. The test is based on the t-distribution, which is similar to the normal distribution but has heavier tails, making it more reliable for smaller datasets. There are three main types of t-tests. One sample t-test compares the sample mean to a known or hypothesized population mean. Independent unpaired two-sample t-test compares the means of two independent groups, e.g., treatment versus control. Paired t-test compares means from the same group measured twice, such as before and after treatment, repeated measures design. The general formula for a t-statistic in a two-sample independent t-test is t equals xx se, where x and x are the sample means of the two groups, and se is the standard error of the difference between the means, which incorporates the sample variances and sizes of the two groups. To apply a t-test correctly, the data must satisfy key assumptions. Normality, the data should be approximately normally distributed. Independence, observations in each group should be independent of each other. Homogeneity of variance, both groups should have roughly equal variances in the case of independent t-test. In biological sciences, the independent t-test is commonly used to compare means from two different populations. For example, one might compare the average heart rate of treated versus untreated rats, or chlorophyll content in leaves exposed to pollution versus clean air. The paired t-test is suitable for comparing measurements taken before and after a drug treatment in the same subjects, such as the growth of plants pre and post-fertilizer application. Once the t-value is calculated, it is compared to a critical value from a t-distribution table, based on the degrees of freedom df and the desired level of significance alpha, typically 0.05. If the p-value is less than alpha, the difference between means is considered statistically significant, meaning it is unlikely to have occurred by chance. It is important to note that the t-test only tells you if the difference is statistically significant, but not if it is biologically meaningful. Thus, effect size and confidence intervals should also be considered for interpretation. When data do not meet t-test assumptions, especially normality or equal variances, non-parametric alternatives like the Mann-Whitney U-test or the Wilcoxon signed rank test are used instead. Previous year question, CSIR Net December 2018. Q. A researcher compared the mean growth rates of two independent plant groups treated with two different fertilizers and found a p-value of 0.03 using a t-test. What does this suggest at alpha equals 0.05? The difference is not significant. B. The two groups have equal means. C. The difference is statistically significant. D. The test is inconclusive. Answer. C. The difference is statistically significant. Practice MCQs. Q1. Which of the following assumptions must be met for a two-sample independent t-test? A. Data should be nominal. B. Samples must be paired. C. Observations should be dependent. D. Variances of the two groups should be equal. Answer. D. Variances of the two groups should be equal. Q2. A paired t-test is most appropriate when A. Comparing the heights of male and female plants. B. Comparing weights of two different species. C. Measuring enzyme activity before and after drug treatment in the same subjects. D. Comparing leaf area in plants grown in different locations. Answer. C. Measuring enzyme activity before and after drug treatment in the same subjects. Let me know if you'd like the next lecture on chi-square test, ANOVA, or confidence intervals. You said. Analysis of variance. ChatGPT said. Here's a detailed, paragraph-style lecture on ANOVA, analysis of variance tailored for CSIR Net Life Sciences, GATE Ecology and Evolution, and postgraduate level competitive exams. This includes theory, biological examples, assumptions, and exam-style questions. Analysis of variance, ANOVA, detailed lecture. Analysis of variance, ANOVA, is a powerful parametric statistical method used to test whether there are significant differences among the means of three or more independent groups. While a t-test can compare means between two groups, ANOVA extends this logic to multiple groups simultaneously, making it a crucial tool in experimental biology, ecology, and life sciences. Instead of comparing means pairwise, which inflates type 1 error, ANOVA evaluates all group means in a single statistical test, based on the variance among and within groups. The basic idea behind ANOVA is to partition the total variability observed in the data into components due to between group variation and within group variation. If the between group variance is significantly larger than the within group variance, it suggests that at least one group mean is different from the others. The core statistic in ANOVA is the F ratio, calculated as F equals variance between groups, variance within groups. A high F value indicates a greater probability that the group means are significantly different. The null hypothesis H in ANOVA is that all group means are equal, while the alternative hypothesis H is that at least one group mean differs. The most common type is one-way ANOVA, which examines the effect of a single factor independent variable with multiple levels, e.g., different diets on a dependent variable, e.g., body weight. For example, a biologist may test whether different soil types affect the height of a plant species. Another example is comparing enzyme activity in response to 3 different pH conditions. Two-way ANOVA is used when there are two independent variables, and it tests not only their individual effects but also their interaction effect on the dependent variable. For instance, a researcher may study how both light intensity and temperature affect chlorophyll content in plants. The assumptions of ANOVA include normal distribution of the dependent variable within each group, homogeneity of variances, equal variances across groups, independence of observations. If these assumptions are violated, especially the assumption of equal variances, then non-parametric alternatives like the Kruskal-Wallis test may be more appropriate. After ANOVA indicates a significant difference, it does not specify which groups differ. To identify where the differences lie, post hoc tests like Tukey's HSD, Bonferroni, or Dunnett's test are performed. These comparisons control for multiple testing errors. In biological sciences, ANOVA is essential when comparing multiple treatments in experiments. For example, to assess whether different concentrations of a pesticide affect insect mortality, or whether different diets influence lipid levels in rodents, ANOVA provides a structured, statistically sound method. Previous year question, CSIR Net Deck 2016. Q. A researcher studied the effect of 4 temperatures on the metabolic rate of a lizard species. Which statistical test is most appropriate to test the hypothesis that metabolic rate varies with temperature? A chi-square test. B. Paired t-test. C. One-way ANOVA. D. Regression analysis. Answer. C. One-way ANOVA. Practice MCQs. Q1. What does the F ratio in ANOVA represent? A. Ratio of standard deviations. B. Ratio of within group variance to between group variance. C. Ratio of between group variance to within group variance. D. Ratio of means. Answer. C. Ratio of between group variance to within group variance. Q2. Which of the following is not an assumption of ANOVA? A. Equal variances. B. Normality. C. Homogeneity of sample size. D. Independence of observations. Answer. C. Homogeneity of sample size. Let me know if you'd like the next lecture on chi-square test, linear regression, or a detailed explanation of post hoc analysis methods. You said, X2. Test. ChatGPT said. Here's a detailed, paragraph-style lecture on the chi-squared test, crafted for CSIR Net Life Sciences, GATE Ecology and Evolution, and other advanced biology-related exams. It includes core theory, assumptions, biological examples, and relevant practice questions. Chi-squared test, detailed lecture. The chi-squared test is a non-parametric statistical test used to determine whether there is a significant association between two categorical variables or whether observed frequencies differ significantly from expected frequencies. Unlike parametric tests like t-tests and ANOVA, the chi-square test does not require assumptions of normal distribution or homogeneity of variance and is especially useful for analyzing count data or proportions. There are two primary forms of the chi-square test. Chi-square goodness of fit test determines whether a single categorical variable follows a specified distribution. For example, it can test if the number of individuals in different phenotypic classes of a genetic cross fits a Mendelian ratio, e.g., 3, 1 to 9, 3, 3, 1. Chi-square test of independence evaluates whether there is a statistical association between two categorical variables, often presented in a contingency table. For example, it can test if the survival of fish is independent of water salinity levels. The formula for the chi-squared statistic is chi-squared equals sigma O, E squared E, where O is the observed frequency, and E is the expected frequency under the null hypothesis. The summation is done over all categories. A large chi-squared value suggests a greater deviation between observed and expected frequencies, indicating that the null hypothesis, no association, may be rejected. The degrees of freedom df depend on the test. For goodness of fit, df equals number of categories 1. For independence in a contingency table, df equals rows 1 times columns 1. The null hypothesis H in the chi-square test always states that there is no difference between observed and expected frequencies or that variables are independent. The alternative hypothesis H is that a significant difference exists so that variables are associated. Chi-square is widely used in genetics, ecology, and behavior. For example, it can test whether offspring ratios in a cross follow expected Mendelian inheritance, or whether species distribution in different habitats is random or shows preference. Key assumptions of the chi-square test include: the data must be in the form of frequencies or counts. Categories must be mutually exclusive. The expected frequency in each cell should generally be at least 5 for the test to be valid. Biological example. In a monohybrid cross of pea plants, you obtain 75 tall and 25 dwarf plants, total equals 100. The expected Mendelian ratio is 3, 1 tall, dwarf. Expected values, 75 tall, 25 dwarf. Observed values, 75 tall, 25 dwarf. Using the chi-square formula, chi-squared equals 75 to 75 squared, 75 plus 25 to 25 squared, 25 equals 0. Since chi-squared equals 0, the observed values perfectly match expectations, and the result is not significant. Previous year question, CSIR Net June 2015. Q. A population of flies segregates for a single gene with 2 alleles. The observed phenotype ratio is 92 red-eyed and 28 white-eyed flies. If the expected ratio is 3, 1, what statistical test should be used to verify this? A. T-test. B. Chi-square test. C. ANOVA. D. Regression. Answer. B. Chi-square test. Practice MCQs. Q1. What is the minimum expected frequency recommended in each cell for the chi-square test to be valid? A. 1. B. 3. C. 5. D. 10. Answer. C. 5. Q2. Chi-square test is used for a comparing means of two groups. B. Testing association between two categorical variables. C. Testing correlation. D. Measuring regression. Answer. B. Testing association between two categorical variables. Let me know if you'd like to continue with linear regression, or would like a comparison of parametric versus non-parametric tests next. You said, basic introduction to mutrovariate statistics, etc. ChatGPT said. Here's a detailed, paragraph-style lecture on multivariate statistics with an introduction and explanation of its applications in biological sciences, tailored for CSIR Net Life Sciences, GATE Ecology and Evolution, and PhD entrance exams. Introduction to multivariate statistics, detailed lecture. Multivariate statistics refers to a set of statistical techniques used for analyzing data that involves more than one dependent or independent variable simultaneously. Unlike univariate, one variable, or bivariate, two variables statistics, multivariate statistics are used when dealing with complex datasets where variables are interrelated, and interactions or patterns among them need to be identified. In biological sciences, ecological modeling, genetics, and behavior studies, multivariate methods help uncover patterns that cannot be detected by analyzing individual variables in isolation. For example, when studying the effect of multiple environmental factors, temperature, salinity, and pH on the distribution of marine species, or analyzing gene expression data where thousands of genes are examined simultaneously, multivariate approaches become indispensable. Some of the commonly used multivariate statistical techniques include principal component analysis (PCA), a dimension reduction technique that transforms correlated variables into a smaller number of uncorrelated components, making large datasets easier to interpret. For instance, PCA can reduce hundreds of gene expression variables into a few key axes representing major variation patterns in transcriptomics. Cluster analysis groups similar objects, species, individuals, or samples based on their characteristics or distance measures. It is often used in taxonomy, microbiome profiling, and community ecology to identify natural groupings or species assemblages. Discriminant function analysis (DFA), a method used to classify observations into predefined groups based on predictor variables. It is widely used in morphometrics, where measurements from body parts are used to classify species or sexes. Multivariate analysis of variance (MANOVA), an extension of ANOVA when more than one dependent variable is tested simultaneously against one or more factors. It accounts for the intercorrelation between dependent variables, which is often seen in biological systems. Canonical correspondence analysis (CCA) and redundancy analysis (RDA), used in ecological and environmental sciences to relate species composition, response variables to environmental gradients, explanatory variables. These techniques provide insights into species-environment relationships. Multiple regression, when multiple independent variables are used to predict a single dependent variable. For example, predicting body weight based on age, diet, and activity level. Multivariate statistical analysis is particularly powerful in the age of big data in biology, such as genomics, proteomics, metabolomics, and ecological modeling. These approaches help to extract meaningful biological information from high-dimensional datasets and reveal hidden structures or interactions. Key assumptions for most multivariate techniques include multivariate normality, absence of multicollinearity, and homogeneity of variance-covariance matrices. Violations of these assumptions may lead to misleading results, so proper data preprocessing and transformation, like standardization and normalization, are often necessary. Biological example. In a study on fish morphology, researchers measure length, width, fin size, and gill surface area. To see if fish from different lakes form separate groups based on these measurements, they use PCA and cluster analysis. PCA reduces the four dimensions into two principal components, and cluster analysis groups the fish into natural clusters, possibly reflecting adaptation to different lake conditions. Practice MCQs. Q1. Which multivariate technique is used to reduce the dimensionality of large datasets? A chi-square test. B. Principal component analysis. C. One-way ANOVA. D. Regression analysis. Answer. B. Principal component analysis. Q2. Which method is suitable for studying species-environment relationships in community ecology? A. MANOVA. B. Linear regression. C. Canonical correspondence analysis. D. Paired t-test. Answer. C. Canonical correspondence analysis. Let me know if you'd like separate, detailed lectures on PCA, cluster analysis, MANOVA, or CCA next. You said. Radiolabeling techniques, ChatGPT said. Here's a detailed, paragraph-style lecture on radiolabeling techniques, prepared for CSIR Net Life Sciences, GATE, and PhD entrance exam preparation in zoology and biology. It includes the theory, biological applications, and exam-oriented highlights. Radiolabeling techniques, detailed lecture. Radiolabelling refers to the technique of incorporating radioactive isotopes, radioisotopes, into molecules to track or quantify biological processes with high sensitivity. These isotopes serve as tracers because their radiation emissions can be detected with high precision. Radiolabelling is a fundamental tool in molecular biology, biochemistry, physiology, ecology, and pharmacokinetics. It allows scientists to follow the path, localization, uptake, metabolism, and interactions of labeled molecules in living systems or in vitro setups. Radioisotopes used in radiolabelling emit different types of radiation: alpha, alpha, beta, beta, or gamma, gamma rays. Among these, beta emitters such as cubed H tritium to the power of 4C carbon-14, cubed squared P phosphorus 32, and cubed to the power of 5S SULFUR 35 are commonly used in biological labeling because they provide stable incorporation into biomolecules like DNA, RNA, proteins, and metabolites. The process of radiolabelling involves either chemical synthesis, where radioactive atoms are incorporated during molecule formation, or enzymatic labeling, where enzymes transfer radioactive groups to specific substrates. For example, cubed squared P labeled ATP can be used to track phosphorylation events in protein signaling pathways, and to the power of 4C labeled glucose can be used to trace metabolic pathways like glycolysis and the Krebs cycle. In molecular biology, radiolabeled nucleotides are used in techniques such as northern and southern blotting, DNA footprinting, in situ hybridization, and DNA sequencing, where radioactive probes allow detection of specific nucleic acid sequences without radiography. In ecology and physiology, radiolabels are used to track nutrient flow in food chains, hormone transport, or the uptake of nutrients and toxins in organisms. Radiolabeling has also been extensively used in pharmacokinetics, where drugs labeled with radioisotopes are administered to animals or humans, and their distribution, absorption, metabolism, and excretion (ADME) are tracked. This provides quantitative data on drug behavior over time. The detection of radiolabeled molecules is typically done using instruments such as Geiger counters, liquid scintillation counters, and autoradiography. Autoradiography involves placing radio-labeled samples on photographic film or phosphor screens, where emitted radiation creates an image corresponding to the location of the labeled molecule. Advantages of radiolabeling include extremely high sensitivity, specificity, and the ability to trace molecules in complex biological systems without disturbing their function. However, the handling of radioactive materials requires strict safety protocols, due to potential health hazards from radiation exposure. Proper shielding, waste disposal, and licensing are essential in labs using radioisotopes. Short-lived isotopes, e.g., cubed squared b, with a half-life of approximately 14 days, are often preferred for short-term experiments, whereas long-lived isotopes like 4C, with a half-life of approximately 5,700 years, are used in long-term labeling studies or for carbon dating. Biological examples. Cubed squared p DNA labeling to detect DNA synthesis rates or to study DNA-protein interactions. 4C labeled amino acids to measure protein synthesis. Cubed H thymidine incorporation to assess cell proliferation in tissue culture. Radio-labeled hormones, e.g., squared to the power of 5I insulin to study receptor binding and signal transduction. Practice MCQs. Q1. Which of the following radioisotopes is most commonly used to label DNA in molecular biology? A to the power of 4C. B cubed squared PC cubed to the power of 5SD squared to the power of 5I answer. B cubed squared B. Q2. What is the purpose of using radio-labeled compounds in metabolic studies? A. To increase reaction rates. B. To visualize and quantify molecular movement. C. To reduce metabolic activity. D. To enhance enzyme activity. Answer. B. To visualize and quantify molecular movement. Let me know if you'd like the next lecture on chromatography techniques, electrophoresis, or spectroscopy methods in biological sciences. You said. Detection and measurement of different types of radioisotopes normally used in biology. ChatGPT said. Here's a detailed, paragraph-style lecture on detection and measurement of different types of radioisotopes normally used in biology, suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology. Detection and measurement of radioisotopes in biology, detailed lecture. Radioisotopes are widely used in biological sciences for tracing biochemical pathways, studying molecular interactions, and quantifying physiological processes. Their utility lies in their ability to emit detectable radiation. To harness their potential, one must understand how to detect and measure the type and intensity of radiation emitted by different radioisotopes. The choice of detection method depends on the type of radiation, alpha, beta, or gamma energy level, half-life, and the biological application. In biological research, the most commonly used radioisotopes include tritium cubed H, a low energy beta emitter, used to label DNA, RNA, or proteins. Carbon-14 to the power of 4C, a beta emitter, often used in metabolic studies. Phosphorus-32 cubed squared P, a high energy beta emitter, frequently used to label DNA or nucleotides. Sulfur-35 cubed to the power of 5S, a beta emitter, used in protein and amino acid labeling. Iodine-125 squared to the power of 5I, a low energy gamma emitter, used for hormone and receptor binding studies. Detection instruments and techniques. Geiger-Müller GM counter. The GM counter detects ionizing radiation by producing a current pulse when radiation passes through a gas-filled tube. It is effective for detecting beta and gamma radiation, especially from isotopes like cubed squared P and squared to the power of 5I. However, it lacks the ability to distinguish between different isotopes or quantify energy levels accurately. Liquid scintillation counter LSC. LSC is highly sensitive and widely used to measure low energy beta emitters, such as cubed H into the power of 4C. In this method, a radiolabeled sample is mixed with a scintillation cocktail that emits light photons when beta particles interact with it. These photons are then detected and quantified by photomultiplier tubes. This method is especially valuable in detecting weak beta emitters that cannot be picked up by GM counters. Gamma counter. Gamma counters use sodium iodide night scintillation detectors to measure gamma rays, especially from isotopes like squared to the power of 5I. This is essential in studies involving radiolabeled hormones or ligands, where gamma radiation penetrates tissue and can be quantified with precision. Autoradiography. This technique uses photographic film or phosphor imaging plates to detect radioisotopes. The radiolabeled biological samples are placed against the film, and the emitted radiation creates a latent image, which is developed to visualize the distribution of the isotope. Autoradiography is widely used in molecular biology, e.g., southern northern blotting and histological localization of radioisotopes. Solid scintillation counting, dry counting. For samples on solid supports like filters or membranes, e.g., in blotting experiments, solid scintillation counters can be used. These utilize phosphor screens or detectors placed directly on the sample. Cherenkov counting. A specialized form of detection for high-energy beta emitters like cubed squared P, where radiation exceeds the speed of light in a medium and produces visible light. This method is useful for counting without adding scintillation fluid, though it's less sensitive than traditional LSC. Measurement parameters. Counts per minute CPM, the raw output of detected disintegrations. Disintegrations per minute DPM, the actual number of radioactive decays, calculated by correcting CPM for detector efficiency. Specific activity, radioactivity per unit mass of a compound, useful for comparing labeling efficiency. Half-life T1/2, the time required for half the atoms of a radioisotope to decay, important for experiment planning and waste management. Biological importance and safety. Detection of radioisotopes allows biologists to quantify dynamic processes such as DNA replication, protein synthesis, ligand receptor binding, and metabolic flux. However, radiation safety protocols are essential to minimize exposure. Shielding plexiglass for beta, lead for gamma, proper waste disposal, and radiation badges for monitoring exposure are standard in all radiological labs. Practice MCQs. Q1. Which of the following radioisotopes requires a liquid scintillation counter for accurate detection due to its low energy beta emissions? A cubed squared P. B squared to the power of 5IC cubed HD to the power of 6 county answer, C cubed H. Q2. Gamma radiation emitted by squared to the power of 5I is best detected using a Geiger counter. B. Autoradiography. C. Gamma counter with Nye detector. D. Liquid scintillation counter answer, C. Gamma counter with Nye detector. Let me know if you want the next topic on RFLP, PCR, or ELISA. You said. Incorporation of radioisotopes in biological tissues and cells, ChatGPT said. Here's a detailed lecture on the incorporation of radioisotopes in biological tissues and cells, written in a paragraph style suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology and biology. Incorporation of radioisotopes in biological tissues and cells, detailed lecture. The incorporation of radioisotopes into biological tissues and cells is a critical technique in experimental biology, used to trace, measure, and visualize dynamic biochemical processes with high precision. This method involves introducing radioactive atoms into biologically relevant molecules, such as nucleotides, amino acids, sugars, or hormones, which then get metabolically or structurally